Welcome to this episode of Mentors at Your Benchside, the podcast giving you advice, tips and tools for getting the most out of your research. I'm Laura Grassi, and today I'll be talking to you about how alkaline lysis works. Alkaline lysis was first described in 1979 and has, with a few modifications, been the preferred method for plasmid DNA extraction from bacteria ever since. The easiest way to describe how alkaline lysis works is to go through the procedure and explain each step. So here goes. Step 1. Cell growth and harvesting. You start with the growth of the bacterial cell culture harbouring your plasmid. When sufficient growth has been achieved, the cells are pelleted by centrifugation to remove them from the growth medium. Step 2. Resuspension. The pellet is then resuspended in a solution, normally called solution 1 or similar in the kits, containing tris, EDTA, glucose and RNase A. Divalent cations, magnesium 2+, and calcium 2+, are essential for DNA's activity and the integrity of bacterial cell wall. EDTA chelates divalent cations in the solution, preventing DNases from damaging the plasmid, and also helps by destabilizing the cell wall. Glucose maintains the osmotic pressure so the cells don't burst, and RNase A is included to degrade cellular RNA when the cells are lysed. Step 3. Alkaline lysis. The lysis buffer, aka solution 2, contains sodium hydroxide and the detergent sodium dodecyl sulfate. SDS solubilizes the cell membrane, while sodium hydroxide helps to break down the cell wall. But more importantly, it disrupts the hydrogen bonding between the DNA bases, converting the double-stranded DNA in the cell, including the genomic DNA and your plasmid, to single-stranded DNA. The process is called denaturation and is a central part of the procedure, which is why it is called alkaline lysis. SDS also denatures most of the proteins in the cells, which helps with the separation of the proteins from the plasmid later in the process. It is important during this step to make sure that the resuspension and lysis buffers are well mixed, although not too vigorously. We'll explain why later. Check out the related article on 5 tips on vector preparation for gene cloning for more information and tips. Also, remember that SDS and sodium hydroxide are pretty nasty, so it's advisable to wear gloves and eye protection when performing alkaline lysis. Step 4. Neutralization. The addition of potassium acetate, normally solution 3, decreases the alkalinity of the mixture. Under these conditions, the hydrogen bonding between the bases of the single-stranded DNA can be re-established so that the single-strand DNA can renature to double-stranded. This is the selective part. While it is easy for the small circular plasmid DNA to renature, it is impossible to properly anneal those huge genomic DNA stretches. This is why it's important to be gentle during the lysis step, because vigorous mixing or vortexing will shear the genomic DNA, producing shorter stretches that can re-anneal and contaminate your plasmid prep. While the double-stranded plasmid can dissolve easily in solution, the single-stranded genomic DNA, the SDS, and the denatured cellular protein stick together through hydrophobic interactions to form a white precipitate. The precipitate can easily be separated from the plasmid DNA solution by centrifugation. Step 5. Cleaning and concentration. Now, your plasmid DNA has been separated from the majority of the cell debris, but it is in a solution containing lots of salt to EDTA RNAs and residual cellular proteins and debris, so it's not much use for downstream applications. The next step is to clean up the solution and concentrate the plasmid DNA. There are several ways to do this, including phenylchloroform extraction, followed by ethanol precipitation, and affinity chromatography-based methods using a support that preferentially binds to the plasmid DNA under certain conditions of salt or pH, but releases it under others. The most common methods are detailed in the article 5 Ways to Clean Up a DNA Sample. So that's it for how alkaline lysis works. Check out the episode description for links to related articles and resources, and don't forget to subscribe to the podcast to get more help and advice from mentors at your benchside.